there is a floor beneath the oldest hardwoods in North America, where a creature with 400 legs spends its entire life recycling death into soil. 12 species, 14 hours, 1 square meter of Appalachian forest floor. And at exactly 7 o'clock, the floor learns to breathe light. But first, the dawn. 5 a.m. to 7:45 a.m. Before the sun has any claim on the sky, the ancient Appalachian forest floor of the Great Smoky Mountains exists in a state of profound stillness. Beneath the canopy, a thick blanket of decomposing hardwood leaves lies in layers that span decades. This is not debris. This is the living foundation of one of the most biodiverse temperate forests on Earth. In the blue-gray hush before dawn, something extraordinary is fading. A male blue ghost firefly drifts centimeters above the leaf litter, its lantern organ emitting a continuous blue-white glow, a cold chemical light generated by the oxidation of luciferin, catalyzed by the enzyme luciferase. Behind its photocyte cells, a reflective layer of urate crystals directs every photon outward. Research published in the journal Physical Review Letters has confirmed that these biological reflectors achieve a luminous efficiency, exceeding 95%, a figure no human-engineered LED has yet matched. It is bioluminescence at its most refined. As the ghost's glow surrenders to dawn, the true sovereign of this floor is already at work. A giant American millipede, Narcissus americanus, a dedicated detritivore, emerges from the shadows. Its hundreds of pale legs ripple in mesmerizing metachronal waves, each diplosement bearing 2 pairs of legs, executing a coordinated motion so mathematically precise that robotics researchers at institutions, including Georgia Tech, have modeled it to improve multi-legged locomotion algorithms. Its Nathocalarium mouthparts scrape and macerate decomposing leaf tissue. What enters as a dead leaf emerges as frass, nutrient-rich pellets that microorganisms convert into the black humus this forest depends upon. Studies in the journal Pedobiologia have demonstrated that millipede frass increases soil microbial biomass by up to 40% in temperate deciduous forests. The millipede does not merely inhabit the forest floor, it builds it. And should a predator approach, tiny ossipores along each diplosegment release droplets of benzoquinone, a noxious chemical deterrent. When chemistry fails, the millipede coils into a defensive spiral, its hard dorsal tergites forming an armored wall. As dawn gives way to an overcast Appalachian morning, a marbled orb weaver, Araneus marmoris, tends her geometric web strung between Christmas fern fronds. Her structural radial threads are composed of dragline silk, a fibroin polymer whose tensile strength exceeds steel by weight, a property confirmed by mechanical testing published in Nature. Nearby, a female acorn weevil drills a borehole through the hard shell of a red oak acorn, using her extraordinarily elongated rostrum. Once the hole is complete, she deposits a single egg deep within the nutritious interior. 7:45 a.m. to 11:30 a.m. A six-spotted fishing spider, Dolomede's triton, maintains her vigil at a stump pool, her hydrophobic tarsal setae dimpling the water surface without breaking through. She hunts not by sight, but by vibration, her Trichobothria mechanoreceptor hairs, detecting the faintest disturbance on the surface tension film. Biomechanical analysis published in the Journal of Experimental Biology has shown that these spiders can detect prey vibrations as small as 1 micrometer in amplitude, a sensitivity rivaling the most advanced piezoelectric sensors. On the bark of a rotting eastern hemlock log, an eyed-click beetle, Alas oculatus, relies on an altogether more theatrical survival strategy. The two enormous false eye spots on its pronotum create an uncanny resemblance to the face of a much larger vertebrate predator. But the click beetle possesses a second, even more extraordinary defense. Should it find itself overturned, a long prosternal spine on its underside locks into a groove on the mesosternum. By arching its body backward, the beetle loads elastic energy into this thoracic hinge. When the spine releases, the explosive force catapults the beetle vertically into the air, often several centimeters, while simultaneously flipping it right side up. High-speed kinematic studies have measured this launch acceleration at over 300 times the force of gravity, one of the highest recorded accelerations in the animal kingdom. The audible crack of this mechanism is one of the most distinctive sounds in the insect world. Beneath this entire surface drama, an even more patient story unfolds. Deep in the Appalachian soil, a periodical cicada nymph, Magicicada septendecim, feeds in perpetual darkness. Its powerful fossorial forelegs, broadened and flattened like shovels, have been excavating tunnels through compacted earth for 17 years. Its stylet mouthparts are embedded in a tree rootlet, drawing dilute xylem sap through a sebareal pump in its head. 17 years of this silent, subterranean patience. Molecular clock analyses, published in Systematic Biology, have confirmed that this extraordinary prime-numbered life cycle evolved as a predator satiation strategy, emerging in such overwhelming numbers that no predator population can consume them all. Through the translucent cuticle of its final instar, the dark crescents of developing adult compound eyes are already forming hundreds of ommatidia, preparing to perceive light for the very first time. As midday approaches, an assassin bug, Pseliopus barberi, completes a lethal ambush. Hanging inverted beneath a fern frond, it seizes a passing insect with raptorial forelegs armed with tibial spines. Its three-segmented piercing rostrum drives through the prey's intersegmental membrane. Through this biological syringe, the assassin injects cytolytic saliva, a cocktail of digestive enzymes that liquefies the prey's internal tissues from within. The bug then draws this nutrient-rich liquid back through its rostrum via the same cybareal pump mechanism employed by the cicada nymph below. It is extra-oral digestion. The prey's own body becomes the vessel in which it is consumed. Proteomics research published in Toxicon has identified over 130 distinct proteins in reduviid saliva, including paralytic neurotoxins that immobilize prey within seconds. (11:30 a.m. to 14:30 p.m.) A stillness descends upon the luminous floor. The flat midday light casts no shadows. The fern fronds hang motionless in the humid air. Beneath the bark, the eastern Hercules beetle lies in diurnal torpor, its massive horn pressed against the ceiling of its refuge. It spotted elytra barely visible in the dim interior. These elytra are not merely protective covers. Research published in Applied Physics Letters has revealed that the cuticle of Dynastes tityus contains a hygroscopic microstructure that actually changes color in response to ambient humidity shifting from olive green in dry conditions to near black in high moisture, a passive camouflage system requiring zero metabolic energy. The marbled orb weaver retreats to her curled leaf shelter, connected to the web hub by a single thread of silk, a signal line that allows her to monitor vibrations while conserving precious energy. Aranid spiders can distinguish between the vibration signature of trapped prey, wind disturbance, and the approach of a potential mate through frequency analysis. Along this single thread, a biological telegraph of extraordinary discrimination. Deep in his moist crevice, the giant American millipede coils in a tight, resting spiral, its spiracles slowly dilating and contracting in the rhythm of tracheal respiration. Respirometry studies have shown that coiled millipedes reduce their metabolic oxygen consumption by nearly 60% compared to active locomotion, a crucial adaptation for surviving the desiccation risk of midday and temperate forests where humidity can plummet beneath the leaf litter surface. And beneath a dead oak leaf hidden from the world, a dormant, synchronous firefly rests with its lantern organ, dark and quiescent, the photocyte cells silent, the luciferin unoxidized, the tracheal network still. The nitric oxide gating mechanism that will later trigger its flash remains inactive, a molecular switch waiting for the signal of falling light. It waits. "30 p.m. to 15 p.m. As the afternoon light warms and softens, the giant American millipede emerges once more from its midday shelter. Its frass pellets now dot the dark humus behind it, visible evidence of the nutrient cycle in action. Isotope tracing studies using carbon-13 labeling have demonstrated that a single population of Narcissus americanus can process up to 15% of the annual leaf litter fall in an Appalachian hardwood forest, contribution to soil carbon cycling that ecologists once attributed almost entirely to microbial decomposition alone. The detritivore is not merely supplementary to the system, it is foundational. The assassin bug repositions on a fresh fern stem, its raptorial forelegs reaching forward into a new ambush. The eyed click beetle patrols the hemlock bark with renewed vigor, its serrate antennae sweeping for chemical signals in the cooling air. Each antenna segment bears thousands of chemosensory scintilla olfactory receptors, so sensitive they can detect volatile organic compounds released by decaying wood at concentrations measured in parts per billion. The cave cricket extends its extraordinarily long antennae from the shelter entrance, sweeping wide arcs through the cooling atmosphere. Neurophysiological research has shown that the Johnston's organ at the base of each antenna processes mechanosensory information with a temporal resolution below 1 millisecond faster than the auditory processing speed of most vertebrates. The handoff from the diurnal world to the nocturnal has begun, and somewhere beneath a dead leaf, the Synchronous firefly stirs. Its photocytes are synthesizing fresh luciferin from the amino acid precursor cysteine. The chemistry of light is loading. 15 p.m. to 19 p.m. And then, as twilight descends upon these ancient mountains, the forest prepares its most breathtaking revelation. A freshly eclosed luna moth, Actius luna, pumps hemolymph through the veins of its crumpled wings, inflating them into 4 vast panels of pale lime-green silk. Each wing bears an eye spot, a dark circle ringed in blue and yellow, while the hind wings trail long, elegant tails that may serve to deflect the echolocation signals of hunting bats. Research published in the Proceedings of the National Academy of Sciences has confirmed that these spinning tails create acoustic interference patterns that redirect bat sonar away from the moth's vulnerable body, an evolutionary countermeasure refined over millions of years of predator-prey escalation. This magnificent creature has no functional mouthparts. It will never eat. Its entire adult existence, perhaps a week, is devoted to a single purpose: reproduction. From beneath the bark, the eastern Hercules beetle, Dynastes tityus, the largest beetle in eastern North America, pushes its horned bulk into the open air. Its pronodal and cephalic horns articulate like pincers, weapons forged by sexual selection for male-to-male combat over mates. And then it begins. A single male synchronous firefly, Photinus carolinus, lifts from the leaf litter. Its lantern organ produces a tentative yellowish-green flash, then another and another. Within minutes, dozens join, then hundreds, then thousands. And slowly, mysteriously, through a process of mathematical entrainment that scientists are still working to fully understand, these thousands of individual flashes begin to synchronize. Computational models published in the Journal of the Royal Society Interface suggest that each firefly adjusts its internal neural oscillator based on the flash timing of its nearest neighbors, a coupled oscillator network, not, unlike the mathematics governing the synchronization of heart, pacemaker cells. 6 seconds of simultaneous light, 6 seconds of absolute darkness, 6 seconds of light. Rolling waves of cold chemical bioluminescence sweep across the entire Appalachian forest floor, like the breath of something vast and alive. The luciferin oxidizes, the luciferase catalyses, the urate crystals reflect, and the ancient darkness becomes a living constellation. Below the waves of light, the giant American millipede continues its patient march. Each flash briefly reveals its dark, armored body legs rippling in their metachronal waves, gnat hocularium mouthparts working, frass deposited behind. It has been here since before the dawn. It will be here long after the last firefly has dimmed. For this is the luminous floor of the Appalachians, where death becomes soil, where soil becomes life, and where, for a few transcendent minutes each year, the forest floor itself learns to breathe light. But there is one question that still haunts the scientists who come here every June. How do they synchronize? How do thousands of individual minds decide to breathe as one? The answer may lie in a completely different forest.